My name is Professor David Jordan. I'm a sorghum breeder geneticist working for the Queensland Alliance for Agriculture and Food Innovation. So lodging is one of the major problems that affects sorghum in Australia. It's a major problem for farmers because the plants, and lodging means that the plants fall over and it makes it very difficult for the farmer to harvest the crop, can lead to large crop losses, reduction in the quality of the product. So when a sorghum plant is subjected to drought, it uses its internal resources to fill the grain, and in doing that weakens the stem, and that weakening the stem causes it to break. And what we're trying to do is to select plants that will be able to deal with this problem. Now the challenge for us is that higher yielding plants are more prone to lodging, and so if we just select for plants that don't lodge, we'll select for low yield. If we select for high yielding plants, then we'll have more lodging. So this is the challenge that University of Queensland, in collaboration with the Department of Agriculture and Fisheries and Advanta Seeds, are trying to solve for Australian farmers.
My name is Professor Andrew Vorrell and I'm a crop physiologist with QAFI at the University of Queensland. So the panicle here, which contains a lot of grains, has a certain demand for carbon and nitrogen. And normally when the leaves are green, through current photosynthesis, they're able to fill the grains. However, if you have a drought after flowering and the leaves begin to die, then photosynthesis is not able to provide the carbon for the grain. In that situation, the plant takes carbon out of the stem and up to the grain to fill it. But in so doing, it produces a hollow stem, which is then much more prone to breaking, and that's how the lodging occurs.
I'm Dr. Geetika. I am a postdoctoral research fellow at Queensland Alliance for Agriculture and Food Innovation, and currently I am working on sorghum lodging. Looking at these differences in stem strength amongst the genotypes and knowing that they are performing better than each other, we would be able to select for these better performing genotypes and eventually be able to breed for this stronger stem, which would result in having a genotype with a high yielding trait and stronger or increased stem strength. Here is the stem breaker. We load our stem onto the stem breaker, which breaks the stem. In doing so, we get a fixed value or the force that was needed to break that stem for that particular genotype. When we look at the data, here on the screen, we get all of these different values of stem strength for different genotypes. And then when we look at this data, I'm able to compare different genotypes with one another, and then be able to say that this particular stem or this particular genotype was actually stronger and performed better than the other. Some of the results that we are already getting is that firstly, we know that there is a wide variation in stem strength amongst the genotypes. And secondly, we think that these differences in stem strength are actually coming from other components such as variations in stem structure, which could be linked to differences in stem diameter or differences in stem shape. One other result that we are getting is that we have found that the stag green genotypes are performing better and they tend to have stronger stem compared to non-stag green genotypes. So I know that my research is solving a question which has been a problem for a number of years for the Australian grains industry and sorghum farmers, and it feels good to know that this research is going to be making a difference for them, as well as increasing grain yields for the grains industry.